I'm Associate Professor Susan Woods. I'm at the University of Adelaide and SAHMRI, and I lead the Gut Cancer Research Group.
And I'm Dr Josephine Wright. I'm a Senior Research Fellow at SAHMRI and I work on gut cancers.
Bowel cancer is one of the most common and deadly cancers. In Australia, over 14 Australians lose their life every day to bowel cancer. So we know for those patients, if we can find the disease early, then we can cure almost all of those patients. So that's really been the focus of our research, is improving early detection for bowel cancers. Like most cancers, bowel cancer affects the older populations in Australia, but what's worrying is that younger patients, so younger than 40, we're seeing a rising incidence in presentation of bowel cancer in those early age groups as well.
This project started when the co-lead of the team from SAHMRI, Dr Dan Worthley, went to visit one of our colleagues, Professor Jeff Hasty, at the University of California in San Diego. And Dan was there for Rob Cooper's lab meeting presentation, where he was discussing a bacteria that he was interested in, which has the ability to take up DNA from the environment and incorporate it into itself. So at this moment, the idea was born, and we wanted to design a bacteria that might be able to take up DNA from the human gut and ideally detect bowel cancer or other gastric cancers. And so using some molecular engineering trickery and CRISPR, we designed and engineered the bacteria to be able to detect early bowel cancer mutations and report on these bowel cancer mutations. So after many experiments and meetings and early morning Zoom calls to the University of California, we have been able to show that we've engineered a bacteria that can detect human tumour DNA and report on this detection.
Meet the bacterium Acinetobacter baylyi. A. baylyi is notorious for finding and stealing DNA from its environment through a nifty skill called natural competence. This allows for horizontal gene transfer so that A. baylyi can integrate cell-free DNA into its own genome to produce new proteins to help it grow and survive.
Our research is focused on the early detection of bowel cancer. We've engineered a bacteria that is capable of detecting human tumour DNA, with the idea that we could deliver this bacteria just like a probiotic, either orally or rectally, and it could detect tumour DNA in situ and then take it up into its genome and report on that tumour DNA by an output signal. The CATCH technology that we've developed stands for Cellular Assay of Targeted CRISPR-discriminated Horizontal Gene Transfer. Now, this mouthful needed an acronym, hence CATCH, but fundamentally it's using a harmless bacteria that is capable of taking up DNA from its environment and using its inherent CRISPR system. We have engineered it to deliver an output signal when it detects human tumour DNA.
We engineered A. baylyi to find and steal DNA, specifically the mutated KRAS gene, which helps colorectal cancer grow. We termed this new bioassay CATCH to collect and characterize cell-free DNA. If A. baylyi found any tumor DNA, it would then turn on a linked antibiotic resistance gene. This tells us that the catch was successful and that cancerous tissue was found. But what does this mean for the real world? This technique of life-detecting life shows how engineered bacteria could be designed to detect specific DNA sequences, to diagnose and even treat disease in hard-to-reach places like, but not limited to, the colon.
In Australia, over 15,000 people are diagnosed with bowel cancer every year, and our standard population screening does an amazing job in finding a lot of those patients early, and it saves lives. However, over 5,000 people still die from bowel cancer, so clearly there are still improvements that can be made to finding disease early. So that's what's brought about our new approach, where we're using live bacteria, to detect life, in this case detecting tumour. And I think our approach and this catch technology is going to be applicable anywhere where we're trying to find specific DNA mutations. And the key for our technology is the bacteria can not only sense that, but because they're a live organism, we can program them to then respond to finding that signal. And that's going to be applicable across lots of different diseases in situations that are really hard to sample. So, for example, in the gut, and we can then bring about a response to that signal.